Okay, you guys, so I'm going to spend some time going ahead and going through this protein synthesis study guide with you guys. So I'm just going to go ahead, I'm going to go through the directions first, and then I will go ahead and we will talk about how to go ahead and do this. Okay, so the first part is just some background information, and they talk about how they were on this planet. They heard a distress call. And now they are going to go ahead and look at what has happened to some of these scientists as well as some of these dogs. So I'm going to start reading this area in here, and then we'll go from there. So it says, I have to keep this quick as I feel the virus making its final push over my mind and body. Dog-like creatures make wonderful pets, but some of them carry a deadly virus. It is unlike anything that we have seen. We don't even know how it's transmitted. Its effects are slow at first, but accelerate once they are past a certain point. We have isolated the source of the virus as it's coming from either the blue or green dog creatures. I'm the last one to succumb to the virus and have started the final DNA analysis to determine which breed is carrying the virus. From there, the computer is set up to create a medicine and a vaccine, and based on the protein that is found. If anyone is reading this, use lab samples A and B to determine which dog is spreading the virus. If the DNA matches that of a specific color of dog, then that is carrying the virus. From there, the computer will create medicine, release it into the air vents of our containment area, and return this to normal. Okay? So those are some of the directions that you guys have there. Your first question is to go ahead and to see how is it even possible to use DNA from an animal to determine its fur color. And we're talking about DNA, protein, amino acids, genes, transcription, translation, nucleus, cytoplasm, base pairs, fur colors, and all of that. Okay. So if you guys remember from the last video, right, the first step that we have is transcription and translation, right? So we have step one is going to be transcription. And you have to remember that in transcription, right, what happens is that DNA goes to RNA, right? And DNA goes to RNA through a process called transcription, right? You have to remember that DNA actually encodes for genes, right? So DNA has genes in it. What happens is that in transcription, we make mRNA and we basically go ahead and we focus in on the gene that we want, right? And since we're focusing in on the gene that we want, right, we're going ahead and we're making this RNA that goes ahead and it takes it out into the cytoplasm. So transcription, one of the big things is that it happens inside of the nucleus. So that is pretty much like one of the biggest things is that transcription happens in the nucleus and we make DNA into RNA. You have to remember that the base pairing rules are slightly different. A goes with U, T goes with A, G is always going to go with C, and C always goes with G. But the big thing is this one right here. This one is the different one that you guys have to be like paying attention to. Right? And then after it goes into transcription, we are going to go ahead and we have our second step, which is going to be translation. And in translation, what happens is that we take this mRNA, right, that is made, and we turn that RNA into a protein. Right, so that's kind of the big thing that happens. And there's a couple of things that happen. So the first thing that happens is that the mRNA is gripped onto by a ribosome. That ribosome goes ahead and it reads the strand, right? So the ribosome reads the mRNA. And then since that ribosome is reading the mRNA, it goes ahead and it builds, right, the protein. So it builds up the protein. And that protein is then going to be expressed. And when that protein is expressed, it's going to go ahead and it's going to show those different traits. And this thing that's going to happen over here is that this also, the location of this, is the cytoplasm, right? So just to kind of give you guys an idea of how this all works, right? So say if this is my cell, right? So if this whole thing is my cell, what happens is that this part in here, right? This is going to be the nucleus, and that's where the DNA is, right? And then all this stuff out here, that is all cytoplasm. So that just gives you an idea of where everything is. Okay, so that's kind of one of the big things on there. And then once you have a protein can express the traits. So those are going to be like all of the different things that you guys have to be able to go ahead and to kind of put into your own words and to be able to really have like a summary of what is going on there. Okay, so I'm going to take some time and show you guys how to decode some of these genes. So I'm going to make sure that you guys know how to do that next. Okay, so the next part is to go ahead and to look at all of these different sequences, and we're going to talk about going from DNA to mRNA. So first, again, I'm going to emphasize the pairing rules, right? So A always goes with U, right? T goes with A, right? And G goes with C, and C also goes with G. So that is pretty much your base pairing rules, right? So what you do is that you're going to go down and we're talking about this mRNA sequence, right? So A goes with U, right? T goes with A, right? C, C, G, G, C, G, U, A, C, G, U, G, C, A, and U, right? So that would be your mRNA sequence. And you're going to do the same thing on the bottom, right? So you go ahead and you write down the letters. Let me grab the eraser. That was really sloppy. So that is a U, a G, a C, U, C, G, A, G, an A, C, A, U, C, A, C, A. Right, so those are going to be all the different letters. And again, I'm following these different pair rules, right? And so the next thing that I'm going to do is that I'm going to go ahead and I'm going to decode the amino acid sequence, right? So again, I'm going to take some time and go through it slowly, and then you guys are going to see me work through this. And then we will go ahead and we'll go from there. Okay, so now the next part is going to be going ahead and determining some of the amino acids that are going to go ahead and to go with it. So in order to go ahead and to determine the amino acid, what we're going to do is that we are going to look at 3 codons at a time. So when we look at 3 letters at a time, that means that we are looking at codons. So just so that you guys can kind of see the pattern, I'm going to go ahead and I'm going to underline the 3 that I'm looking at a time to kind of keep track of it. And then we will look at the codon chart and we'll determine from there how to go ahead and to find it. So the first one that we have is UUA. And I'm going to show you guys on both codon charts how to do it. Lots of students like the circle one, so I'm going to use the circle one first, and then we'll go through the rest. So for this one, we have U, so I'm going to take this big U, right, and then this next one that's kind of shaded in, that's going to be your second one. So I have U, U, and then I have A, right? So U, U, A, that means that is going to be leucine. So I would go ahead and I would write leucine. And it's the same thing on here, right? Oh shoot, this is a bad chart. So it'd be the same thing on a chart that looks something like this. Let's go ahead and let's look at this chart. Open link in new tab. So if you look at this, right, we go U, U, and A. So U at the top, because that's your first codon letter, right? So this is the first letter. So first letter is here. So I have U. My second letter is going to be here. And then my third letter is there. And then that's the same thing. It codes for leucine, L-E-U. So when we go back to our thing right here, UUA codes for leucine. So L-E and U. And you guys can't really see that well. So I'm going to change the color. Right, so that's going to be leucine, right? The next one is AAC. So you guys are going to look on here, and it's going to be A, and C, which is asparagine. And you guys look on here as, not this one. I'm going to delete that tab. Then you guys look on here, right here, it's A, and G. Whoopsies. Wait. Go back here. ACC, my bad. So you're going to go ACC. So A, C, C is theanine, right? Because that's where it all collides. Oh no, that's not right. C, asparagine. There it is. Now I got it right. My bad, guys. So it's going to be A here, right? So A, C, right? And because that is there, that's going to be asparagine. And we got the same thing on here. C, asparagine, right? So N. So we're going to go ahead and we're going to write that there. So it's going to be ASN, right? And then we go GGC. So GGC. So G, and C is going to be glycine, GGC. So we'll double check on here. GGC is glycine. And you guys should get the same letters no matter which chart you use. So whichever chart gives you the right letters more often, that's going to be the chart that I would recommend using for the test, right? So next, GUA. So I'm going to start using this chart. GUA is valine. So I'm going to go ahead, I'm going to write valine on there. Then I have ACG. So I have ACG, theanine, so THR. Theanine, UGC, UGC, cysteine, and AUU. So AUU is isoleucine. So that's going to be the letters on here. And you're going to go through the same process down here. So you're going to pair them off into threes. And then you guys are going to go ahead and to determine, right, what they code for. And then there's one more step after this. So UAU. So UAU is threonine, so TYR. GCC. GCC is alanine. UCG. UC and G is serine, so SER. All A's, so A is lysine. GAC. GAC is aspartic acid. Oops. AUC. AUC is isoleucine. And ACA. Oopsies. ACA is threonine. Okay, so now the next step is to go ahead and to determine, right, what color are these animals going to be and what they go ahead and what they code for. So now what we're going to do is that we're going to scroll up a little bit and we are going to look at some of this stuff right here, right? And we have to see if any of our sequences match any of these colors, right? So I have threonine for none of them start with leucine, so I don't think I have a color for dog for that. But I do have this threonine, so this TYR. And then I have ALA. Then I have SER, LYS, ASP, which is aspartic acid, isoleucine, and THR. So I go ahead and I look at that and I see that those match completely, right? So the color of this dog right here is going to be blue, right? So the color of this dog is going to be blue. And then this one right here, since this code does not match any of these things right here, right? This means that this animal does not have the virus, right? Because if we read before, right, it mentions that if it doesn't code for a color, it does not have the virus. So this one right here does not have the virus, right? The one that at the bottom that matches, right, the blue one, it does have that virus, right? And we know that it has that virus because the codes for the proteins match. Okay, so I wanted to give you this just to kind of overview some of the skills that you guys will need, and then we will go ahead and we will wish you guys the best of luck as you guys try out your first performance task.